Point mutations. Point mutations are those mutations that affect the single site of the DNA. They can be of two types. They are called as substitutions, or the second type is called as addition deletion, which result in the form of frame shift. Transitions. If purines are replaced by purines and pyrimidines are replaced by pyrimidines, then the mutation is called as transitions. While on the other hand, if purines are replaced by pyrimidines and pyrimidines they replace the purines, then this phenomena is called as transversions. Point mutation results in three forms. It can be in the form of silent mutations, in the form of missense mutations, and in the form of nonsense mutations. But in addition to these three types, if there is single nucleotide deletions or single nucleotide insertions which result in the form of frame shift mutations, they are also considered as types of the point mutations. In this diagram, we can see that what are the point mutations. This is the normal DNA, RNA and protein sequence. Point mutations, if there is a mutation at one point, mean C is replaced by T, then it produces lysine. In the original sequence, there was lysine. Double TC was producing lysine, was responsible for the amino acid lysine, and if there is a change in place of C, now here is T, but this codon also codes for the lysine. So if there is change at the DNA level but there is no change at the protein level, then it is called as silent mutation. While in the next example, if there is a change at A, originally there was T, instead of T, now here is A, and as a result of this point mutation, the amino acid is converted into stop codon. So this type of the point mutation is called as stop codon, where there is a stop for the formation of the amino acids or the polypeptides. The third type of the point mutations is missense mutations. In case of missense mutations, in the original sequence here was T, but in case of T, now there is C, and in this case, which is also missense, instead of T here is G. So the original amino acid was lysine, and in this case arginine, and in this case it is threonine. So because the lysine amino acid is substituted in this case by arginine and in this case by threonine, if one amino acid is replaced by another amino acid, the phenomena is called as missense substitution. So point mutations can be of three types: silent mutation, nonsense mutation and missense mutation. In case of silent mutation, there is a change at DNA level but there is no change at protein level. In case of nonsense mutation, there is a change at DNA level, but due to that change, there is a stop for the formation of the amino acids or of the polypeptides. In case of missense mutation, one amino acid is replaced by another amino acid. So point mutations are those mutations that affect the DNA at a single point. They can be in the form of silent mutation, missense mutation, nonsense mutation. They can also be in the form of small deletions and insertions, which ultimately result in the form of frame shift mutations.